Okay, we're live. So good morning, everyone, and welcome to this webinar about fluorescence lifetime imaging microscopy and more precisely about the Lambert instrument. We wait 2-3 more minutes just to make sure that everyone is here, and then we'll start. In the meantime, if you write something right now. Just let us know if you have any issues, if you can't hear us or anything like that. It should be okay. So. Okay, let's maybe wait 1 more minute. Hi, Celeste. Okay, so let's start. So again, good morning, everyone. Welcome to this webinar about fluorescence lifetime imaging microscopy, and more precisely about the Toggel FLIM system from Lambert Instruments. I'm Vincent Renaud, Sales Application Engineer at Axiom Optics, and here is also Jasper Bosch from Lambert Instruments, Development Engineer at Lambert Instruments. You can't see him right now, but you will be able to see him very soon. So I will start with a very quick introduction of Axiom Optics. Axiom Optics is the distributor of Lambert instruments in North America. We specialize in optical instrumentation. For the microscopy division, we are a team of 3 people. Philippe, Neche, and myself. Philippe and Neche are on the East Coast, and I'm myself based on the West Coast. In microscopy, we specialize in microscopy add-ons. So for instance, we offer the Rescan confocal microscope, which is an add-on that transforms a wide field system into a laser scanning confocal system with super resolution capability. We also offer the Mikao adaptive optics add-on for single molecule localization microscopy and single particle tracking. Of course, we offer the Toggel FLIM system from Lambert Instrument, which is the topic of today. And then recently we started working with Imperax. We offer the SensoCell optical tweezers and force measurement platform. Besides optical system, we also offer cameras and tools. We have a wide variety of cameras from CMOS for, I would say, very regular microscopy applications. The Tuxen CMOS cameras are 20 megapixel, very low noise cameras and really inexpensive, 2200 USD. We also have more advanced CMOS cameras for more advanced applications requiring more sensitivity and also more speed. So the Orca Flash 4, Orca Fusion, and Orca Lightning camera. And when we're talking about speed and sensitivity, we of course have to mention the HiCam Fluo camera from Lambert Instruments, which is an intensified CMOS that can go at 1000 FPS for 1.2 megapixel, and is also very sensitive, so very good for applications requiring both speed and sensitivity, like voltage indicators, etc. Beside cameras, we also offer the quality control solutions from Argolite, which are a very good tool for any microscopy facility to measure pretty much everything on the microscope, from resolution to excitation intensity, homogeneity of the field of view, etc. So this is done for the introduction of Axiom Optics, and now I'm going to give the hand to Jasper, who is going to kick off this FLIM webinar and tell you more about fluorescence lifetime imaging microscopy and about the Toggel FLIM system. And I will see you for the question and answer at the end of this webinar. Jasper, it's for you.
Thanks, Vincent. Thanks for that very nice introduction. And thanks to everybody for joining us today for this FLIM webinar. So my name is Jasper Bosch. I'm a development engineer for Lambert Instruments, and I've been working on the Toggel camera and our FLIM systems for, well, ever since I started working here. So let me tell you what I wanted to discuss today. I'll start with a brief introduction of Lambert Instruments, and then we'll go over the basics of fluorescence lifetime imaging theory and the applications of fluorescence lifetime imaging. Then we'll go on to discuss frequency domain FLIM and why it's such a nice way to do FLIM measurements. And then we'll dive into the software and see how the system works, how you can analyze your data. And then at the end, there will be time for questions, of course. So let's start with a short introduction about Lambert Instruments. Lambert Instruments is a small company in the Netherlands. We're on the top floor of this building that you see in this image. But we have customers around the world because we have a lot of experience in low light imaging and fluorescence imaging. So those are our two core expertises. And we make cameras that are extremely sensitive to light, and we make cameras that can measure the fluorescence lifetime from the usually quite weak light that comes from fluorophores. And for these kinds of applications, we develop complete solutions, which can include camera, a light source, and software, and make sure that everything works together so that these challenging experiments that they're used for work great. So on to fluorescence lifetime imaging. To start at the beginning, I'm quite sure everybody knows this, but fluorescence is when you shine light on a molecule and that light is absorbed by the molecule, and then after a while it's re-emitted by the molecule. So first light shines on the molecule goes into its excited state, and then after a while it will re-emit the excess energy in the form of fluorescence light. And it's that fluorescence light that we're looking at with our FLIM systems. And this fluorescence light is always, of course, a longer wavelength than the excitation light, because the emission photon has less energy than the excitation photon. So because this is a probabilistic process, this re-emission, it always happens at the same time since the excitation. So if you shine light onto the same molecule a couple of times, it won't always take the same amount of time for that molecule to re-emit the light. You'll see that it has an exponential decay curve. And it's the slope of this exponential decay curve, which is the time constant tau, that quantifies the fluorescence lifetime. It's that quantity that we image with our FLIM systems. So why would you want to do this? This is, of course, a very important question. And the reason is that using fluorescence lifetime imaging and by measuring the fluorescence lifetime, you can extract incredible information from your samples. You can get information from living samples that you would normally not be able to get. So one of the applications is in neuron imaging and in brain research, where where researchers use green fluorescent protein and M-cherry and look at where they interact. Because when you have the green fluorescent protein and the M-cherry and they interact, that tells you that there is protein interaction in the synapses. And you see that in these 3 images in the bottom right corner of the screen. If you're just looking at the green fluorescent protein, you'll get the image on the left, and it has a longer lifetime if there's no M-cherry that's interacting with it. And what you'll see on the right image is that there is mainly interaction in the synapses. And you see this by looking at the colors, because the colors in this image, they are a pseudo-color overlay of the fluorescence lifetime. And what that means is that the color that's overlaid tells you something about the lifetime. So red is a longer lifetime and green is a shorter lifetime. Another application that FLIM is often used for is live cell imaging in biomedical applications like cancer research. Now, in such situations, the researchers use fluorophores that are sensitive to a specific environmental parameter. This can be the pH level, or the oxygen concentration, or the calcium concentration. So let me show you a time lapse that was recorded by one of our customers at the Netherlands Cancer Institute. And let me just switch to that now. I'm not quite sure if this is working.
You need to go to video.
Yeah,
and then to start the video.
Do I need to
the
stop
HeLa cells?
Yeah. I need to stop the presentation first, I suppose.
It's playing now.
Yeah, okay, it's playing now. Okay, sorry for that. So in this video, what you see is that there are, oh, now it's gone.
Let's play it again.
Yeah, let's play it again. So after 2.5 minutes, the researchers added isoproterenol to this sample. And what you see is that the lifetime of each cell changes quite rapidly. Let me just show that one more time. And you see this by the color changing from blue to green or orange, and then it changes back again, which means that for a very brief moment, the let me get the slides back. For a very brief moment, the lifetime of the cells change, which means that there is a rapid increase in the concentration of cyclic AMP in those cells, which is very important for biological processes where it is a second messenger. Another application is high throughput screening. So sure you're familiar with these 96 well plates, and in this specific application, the video that I'm showing, the image that I'm showing on the right, this is from the research of the University of Amsterdam, where they developed a red fluorescent protein called M-scarlet. And while they were developing it, they of course tried many different types of protein. They tried a lot of variations of the protein, and they used our FLIM systems and a motorized microscopy stage to image each of the wells, and each of those had a different variant of the protein in it. And by doing it this way, they were able to very quickly assess all the different variants and find the one that had the best properties, the properties that they were looking for. And you can read about it in the publication that's also referenced at the bottom of this slide in Nature Methods. And then just one other application I will mention here is microfluidics. So in microfluidics, this customer was studying the rates of gas absorption into water. And so they they dissolved oxygen, and they were looking at the fluorescence lifetime, because they were using a luminescent dye called ruthenium tris dichloride hexahydride. Okay, I'm sorry for my pronunciation of that. But this dye changes lifetime for a different oxygen concentration. So by measuring the lifetime, you can actually use it to read out a physical property in your sample. In this case, the oxygen concentration. So in general, that's what FLIM is often used for, to read a physical property of your sample using the light that is emitted by the sample without further disturbing your sample. So your sample can be in its natural state, your cells can be in their natural environment, but you can read the information that you want to know about the cells. Okay, so let's move on to frequency domain FLIM. Frequency domain FLIM is one of the ways to measure the fluorescence lifetime. There are two main ways to measure it: are time-correlated single photon counting, which is often used on confocal microscopes, and there's frequency domain FLIM, which is the method that we use in our products. So one of the nice things about frequency domain FLIM is that we just have a camera connected to the microscope. So in the image on the right, you see our FLIM camera. On the left is connected to the camera port of the microscope. And on the right, there is a light source. It can be a modulated LED light source, or it can also be a laser light source, depending on what your application needs, of course. And this system also works on spin disk confocal and on total internal reflection microscopes. So how does this work? The light source emits light which is modulated, which means that the light intensity is varied in a sinusoidal way. It's basically switched on and off very quickly. This is always at megahertz frequencies if you want to measure nanosecond lifetimes. This light shines onto the sample where it is absorbed, and the sample then re-emits it at a longer wavelength. Now this is the light that goes to the camera. So we collect the fluorescence light with the camera, and we record the light intensities that the camera sees. And because both the light source and the camera are modulated, we can change the phase between the light source and the camera. So if the light source and the camera are in phase, then the camera will be recording light whenever the light source is on. So the camera will record a lot of light, and that will be recorded as one image. And in the graph on this slide, that would be one of the green dots at the top of the sine wave. And then we shift the phase between the light source and the camera. And what we see is that if we shift the phase, the camera will record a different amount of light. And so we shift the phase in steps all across the 360 degrees to collect light at each of those phase steps. And then we can fit a sine wave through that. And we can do this really quickly. So each pixel gets a reconstructed sine wave that contains all the information that we need to calculate the lifetime. To then get the lifetime of your sample, we need a recording of a reference slide with a known lifetime. Because from that known lifetime, we can then see how that relates to the sine wave that we recorded of the sample, and we can compare it to the reference sine wave, and then we can calculate the exact lifetime of your sample. If you want more information about how this calculation works, you can find that on our website at lambertinstruments.com/flim-equations. Or if you have any further questions about how this works, you can also ask during the Q&A after the presentation. So some of the advantages of frequency domain FLIM. Now, like I said, we use a camera to record the image data. And the good thing about this is that it works very easily. Everybody is familiar with using cameras nowadays, and it's just very easy to use. It's also very robust. With the camera, there are not a lot of things, not a lot of moving parts that can break. So it's a very robust system, and you can have a novice user work with it within about 30 minutes. So it's very easy to work with. It's also really fast. Acquiring one FLIM image, once you have the reference slide measured, recording one image of a FLIM sample, it takes just a matter of seconds, or even less than a second if your sample is bright enough. And then after you have recorded the image data, the lifetime calculation happens instantaneously. So there's no fitting, there is no adjusting settings. It just calculates the lifetime instantly and shows it to you. And during time lapses, you get live updates of your lifetime, and you can see everything that's happening in your sample in real time. And because we're using a camera, you're seeing a large field of view, so you can see a large part of your sample. And if you want to zoom in, you can just use a different microscopy objective to get a really zoomed in image of your sample. Another advantage is that all pixels are recorded simultaneously. And this is different from time-correlated single photon counting, because in time-correlated single photon counting, not all pixels in your final image were recorded at the same time, because they are recorded by scanning the sample row after row and column after column. And that means that in your final image, the pixel in the top left of the image is not recorded at the same moment as the pixel in the bottom right of your image. But with frequency domain FLIM, because we're using a camera, each pixel is recorded simultaneously. And that means that there is no motion artifacts. Even if you have cells that are moving around a bit, you won't have any motion artifacts in your images. There's also no pileup effects. With TCSPC, you can have pileup if a part of your image is far too bright, and that can show up by showing a lifetime that's much shorter than you would have expected. But with FLIM, with frequency domain FLIM, you don't have that problem. Another advantage is that a frequency domain FLIM system is affordable. In general, it's much more affordable than a time-correlated single photon counting system. If you want more information about that, contact the people at Axiom Optics if you're in the US, and otherwise you can contact us at Lambert Instruments directly. So much for the theory. Let's now go into the demo section of the webinar. And we will do that by going into our software. And I will share the screen of our software with you. So unfortunately, we had a technical issue with the camera earlier this afternoon, despite it being very robust. And because of that, I don't have a camera to show you live image acquisition. But what we can do is we can move through the software, and I can describe how everything works, how fast everything goes, and then we can look at the pre-recorded data that we have and do the analysis on that. So let's start by an overview of the interface. So this is the first screen that you'll see when you start the software. On the left, you have all your settings, and then right next to it, you have the image of the camera. At the bottom of the screen, you have a statistics table that contains the statistics of your samples, but we'll get to that later. So on the left are the settings for our FLIM measurement. We can enter the reference lifetime of the reference slide that we're measuring. So this is where we would enter the lifetime of the reference slide that we would be using. So if you're using fluorescence, you would be entering 4 nanoseconds here. Or if you're using erythrocene, you would be entering 86 picoseconds. And then that will be saved to the recording of your reference slides that you would be making. We can set the exposure time of the camera and adjust it so that the image just has just the right exposure, just has a nice amount of light, so you can really see all the details really well. And we can set the phase. Now this phase is the phase difference between the light source and the camera. And by varying it, you're able to see the change in light intensity that I just described before. So the light intensity changes as you change the phase between the light source and the camera. There's also a gain slider, so you can increase the CCD gain of the camera to achieve higher recorded values. And there is the modulation frequency of the camera, which is also then set for the light source. So whatever modulation frequency you select over here will also be applied to the light source. And then there is number of phases. So normally we recommend using a 12-phase acquisition. So we're shifting the phase in steps of 360 degrees divided by 12. So that's 30 degree steps. And then we've got 12 points to reconstruct the sine wave. But you can also set a higher number if you want to make sure that your lifetime is as accurate as possible. Or you can set a lower number of phases. So if you have a sample that's moving very quickly, or if you have a sample that maybe bleaches very quickly, then you can use a smaller number of phases, which could also be helpful if you want to see very fast changes in lifetime. And then there's the multi-LED settings. So these are the settings for our multi-LED product, which can have up to 4 wavelengths of LED. And it works very simply. You just select the wavelength that you would like to use. And then when you start a recording, the shutter of the multi-LED will be opened automatically. Now, let me tell you how a normal reference slide acquisition would work. You would put the reference slide on the microscope, of course, and set the exposure time of your camera, select the modulation frequency that you would like to use, and then you would click Record Reference. Now, depending on the number of phases that you've entered, and of course, the brightness of your sample and the corresponding exposure time of the camera, recording a reference measurement takes about 1 or 2 seconds after you've clicked Record Reference. So that goes really quickly. And once you've recorded a reference, you can use it all throughout the experiment session that comes afterwards. So in this slide that you see over here, in this image, we've recorded a reference of a fluorescent plastic. And that has a lifetime of 5 nanoseconds. So once you've recorded your reference slide, you can move on to doing your experiments. And to do that, you go to the Acquisition tab. So in the Acquisition tab, you can again set your exposure time and your gain and your modulation frequency and number of phases. And then you can click Record Sample. Now, recording a sample takes about the same amount of time as recording a reference, but it all depends on how bright your samples are. If your sample is very bright, then you can record multiple fluorescence lifetime images a second. You can achieve a speed of 2 frames per second, 2 lifetime images per second. Now, the lifetime images are presented with a pseudo-color overlay that you see on the right here. So let me just remove that region of interest. So on the left, you see the light intensity that was recorded for this, this plant sample. And then on the right, you can see the color overlay on the light intensity. And again, the pseudo-color in the image tells you something about the fluorescence lifetime in that part of the image. So this part of the root is very blue, which means that it has a shorter lifetime. It has a lifetime at the bottom end of the color bar on the right side of the screen. And then the different parts on the right and on the bottom, they have longer lifetimes because they're yellow and red. So we can draw a region of interest, and then the information for that region of interest is shown at the bottom of the screen. So we can see that the lifetime of the blue part is around 2 nanoseconds, and the yellow and red part is around 4.5 nanoseconds. So that's how recording a single fluorescence lifetime image works. And if you were to walk out of your lab to your microscope and put your slides on the microscope, do a reference recording first maybe. You can all do this within a matter of just a couple of minutes. If you're experienced with the system, I would say that you could do this in 2, maybe 3 minutes, and then you would have your lifetime measurement for your sample. So it's really quick and really easy. Now, what most customers use a lot is the time-lapse feature. So before I spoke about recording a single fluorescence lifetime image, but the software can also record multiple lifetime images that are recorded one after another with a specified interval between them. So that's also all shown in this panel on the left of the screen. You can enter the duration of your experiment. So let's say that you would like to run your experiment for 6 hours, then you just enter it over here, and you can set an interval. So if you want the interval between the start of each fluorescence lifetime acquisition to be 10 seconds, then you would set it like this. If you want to start a recording every minute, you would set it like this. And then you would simply press start time lapse. If you want to record the next FLIM image right after the previous one is done, you simply click minimize, and it will calculate the minimum time that it takes to record the data and then enter that. So you can go back-to-back FLIM imaging. So then once you've recorded your time lapse, you can analyze it in the analysis tab, which is also where you can analyze single fluorescence lifetime images, of course. So to show you this, I'm going to load a pre-recorded time lapse that was recorded by one of our customers at the Netherlands Cancer Institute. Let me just run the recalculation real quick. So let's start with the fluorescence lifetime overlay again. And I can change the pseudo colors in the image by changing the limits of this color bar. So right now, the fluorescence lifetime of 6 nanoseconds will be shown as red, but there's no red in this image. So we can change the scale of this color bar by entering a different value. So we can enter 4, and you'll get some red over here in your image. And you can just set the pseudo color to whatever value you like to get a nice image. And these limits of the color bar are then also applied to the limits of the graphs on the right side of the screen. So right now we're looking at the histogram of the fluorescence lifetime that's calculated from the phase of the sine wave. We can also calculate the fluorescence lifetime from the modulation of the sine wave, but we'll get to that later. So if we're looking at this histogram, we can see that most of the lifetimes are between 2 and 2.5 nanoseconds. But what happens during this time lapse is that a substance is added, isoproterenol, and the lifetime changes. So as I move through the time lapse, you'll see the histogram update as well. And this would also happen during the acquisition of the lifetime data. So I can press play on the player below, and then the time lapse will be played back. And keep an eye on the histogram on the right side of the screen and see how it shifts to the right and then back to the left again as the cells go back to their start lifetime. We can also draw a region of interest around a specific cell to see the histogram for just that cell. And we can draw a region of interest around different cells and then switch between them to see how their histograms look. So for a time lapse like this, it could also be interesting to look at how the lifetime changes over time. And to look at that, we can select a different type of plot. So right now we're looking at the histogram, but maybe we want to have a look at the lifetime versus time. So I just select that over here, and we can see that the lifetime starts out between 2 and 2.5 nanoseconds. Then it increases to almost 3.5, and then it goes back down and decreases slowly after that. You can also have multiple plots simultaneously. So let's say that I want to show a histogram of the fluorescence lifetime and simultaneously a scatter plot of the lifetime versus time. And again, these are all rendered in real time during time-lapse acquisition as well. So you can really see your data build up. Another important type of plot is the polar plot or phasor plot. So this type of plot is calculated from both the phase and modulation information that is in the FLIM data that we record. And it tells you about whether or not the lifetime information is mono-exponential, and it tells you about the magnitude of the lifetime as well. So what you'll see here as we move through the time lapse again is that you see this blob over here in the polar plot, and these pixels correspond to the pixels in the region of interest number 5 that I've drawn in the image. So each of these coordinates in this plot correspond to a specific phase and modulation in these pixels of this cell. And as the phase and modulation change, you'll see that the blob also moves within the semicircle. So let me just play back the time lapse, and during the time lapse you'll see the blob move as well. So you see the blob move sharply to the left when the isoproternal is added, and then it moves back slowly as its lifetime increases again. And for each of these cells, that trajectory might be different. So cell number 4 might move back more slowly than cell number 5. And these are the kinds of analysis that you can do with polar plots. So let me show that you can also have 4 of these plots simultaneously. So you can really get a lot of data showing during your acquisition as well. And all this information from the cells that you've selected, let me select a couple more. You can also use a freehand region of interest if you want to draw a specific shape around the cells. All this information then appears in the statistics table that's at the bottom of the screen on the right. And you can switch which region of interest you want to show in your plots by clicking on the check mark in front of it. And then once you've got all the data that you want to collect, you can simply select all of these, and you can just copy and paste them right into Excel or any other type of spreadsheet program that you use by pressing Control-C, Control-V. If you want to export the information from any of these plots, simply right-click on it, and you can either save it as an image, so you would then have the image that's showing right now, or export data to get the raw data that's showing in this graph, so you can maybe use it in a graph that you want to make yourself. So that's a quick overview of the LIFA software and a quick overview of the LIFA fluorescence lifetime imaging workflow. And with that, I want to go back to the presentation. And oh, yeah. So we're going back to the slides. Okay. So that was the hands-on demo part of the webinar. As you saw, the The LIFA software controls both the light source and the camera, so all your hardware is connected in the same software, but maybe you want to use a different platform to do your experiment. And for such users, we have support for Micromanager and other platforms like Python, LabVIEW, or MATLAB. So if you want to build your entire setup in Micromanager, then you can also get the toggle and the multi-LED in there and automate your full experiment that way with the other equipment that you might have in your lab. Now, you might have seen during the presentation that in the time-lapse panel, there was also a single image FLIM or SciFLIM option. And this is based on a smart trick that the sensor in this FLIM camera does. And it's that two images are recorded simultaneously, but with opposite phase. And from this information, you can then calculate the lifetime as well. Now, the advantage of this is that it's very fast. So in this plot on this slide, you can see that you can see the lifetime changes between 2.8 and around 3.3 nanoseconds. And these happen very quickly. And with a normal TCS-PC or not so fast FLIM system, you would not be able to see these changes in lifetime, these very fast changes, especially if you look to the plot on the right side of the screen that shows fluctuations between about 3.2 and 3.3 nanoseconds. And these are very fast. There's about one fluctuation every second. So with the toggle, these researchers were able to record 20 FLIM images per second using the single image FLIM method, which is also implemented in our software. If you want to know more about single image FLIM, you can have a look at lambertinstruments.com/cflim. And I can show you the video of these fast calcium spikes right now. So let's see if that works. I think it's not showing. Play. Yeah, okay, here we are. So a substance is added to these cells as well, and then you can see really fast calcium spikes in this sample as well. So let me go back to the slides. So that's an example of how fast you can do fluorescence lifetime imaging with this camera. So with a normal time lapse, where you record multiple phase images for a lifetime image, for a lifetime calculation, you can get up to around 2 lifetime images per second. Or if your cells are very bright, you can go faster than that. And with single image FLIM, you can go even a lot faster, up to 30 fluorescence lifetime images per second. But you would have to have very bright cells to make sure that there's enough light coming from your cells to record a good image. So that brings us to the Q&A part of the session. I think Vincent will join us again for this.
Yes, I'm back.
Hi, Vincent.
Thank you so much, Jasper. Thank you so much. So yeah, we have several questions. I think I can answer most of them, but I will need your help sometimes, Jasper. So the first question is from Days. Is frequency domain FLIM a wide field acquisition method? So let's discuss about the toggle. The toggle is a camera-based system. So yes, it works in wide field, but it also works in other techniques which are camera-based. So for instance, spinning disk would work, turf would work, light sheet microscopy would work as well.
Yeah.
I think it doesn't tested in spinning disk and turf, right? Correct?
Yeah, that's right. Yeah.
So pretty much everything, as long as it's using a camera, would work with the system.
That's right. As long as there's a camera port on the system, you can hook up the this FLIM camera to it and get some pretty good fluorescence lifetime images. Yeah, definitely.
So I will jump to another question, which is I think related to that as well, which was about the excitation light source. So we need to use a modulated light source. Let me find the question. It was a bit after. Yes, from Bo. So how do you modulate a light source? I notice the modulation frequency reaches 20 megahertz. That's pretty fast. So yes, that's correct. It's pretty fast, and not all excitation light source can do that. Lambert instrument offer the toggle camera combined with light sources. So for instance, the multi-LED, which is basically LEDs, or a multi-laser, which is laser-based, mostly laser diode. You worked with, I mean, you already have some wavelength that will work easily, and you also work with a laser company. So if you already have a system, we need to discuss if it would be compatible or not. Anything to add, Jasper, or was it good?
I think that's a very good question, and you answered it perfectly. So our multi-LED can go up to 60 megahertz, and if you go any faster than that frequency, the LEDs are starting to be just not fast enough anymore to switch on when you modulate at a speed like that. But up to and including 60 megahertz, that works really well with our multi-LED. And then of course with the lasers, they can modulate at even higher frequencies, and they have excellent modulation depths and light intensities. So those are also great.
Perfect. And maybe it's also good to mention that it's a safe bet that any laser diode would be able to be modulated that fast. But if someone is using a LED for AP fluorescence, for white field illumination, there's also a chance that it might not work, right? You're using special LEDs, correct?
We are using special LEDs, that's right. And we're also using our own custom LED driver electronics to drive the LEDs at these speeds. So you do need the specialized hardware to use LEDs at these frequencies. Yeah.
Okay. Also related to this question, the question from Silas, does the system return just a single lifetime value for each pixel, or does it calculate the distribution of lifetime decay values? So I think the confusion comes from the fact that we're taking 12 images to get one lifetime. And it's actually part of the process to calculate the lifetime, but it doesn't mean that we are acquiring 12 different lifetime. With 12 images, we can calculate one single lifetime. So the answer to that is, Silas, is we get one lifetime per pixel for one acquisition.
That's right. However, you do get the phase and modulation information for each pixel. And from both of those, you can calculate the lifetime. So you're actually getting 2 lifetimes from each pixel from that sine wave that you are measuring. Yeah.
Perfect. Thanks, Jasper. I couldn't. I don't know how to answer to this question from Neil. Do you have data on the variation of SNR of frequency accuracy across the image sensor?
That's very specialized technical question. Do we have data on the variation of the SNR?
Or is there maybe a variation of the SNR over the sensor?
Mm-hmm. Yeah, we do have that information. We also measure it, of course, during the testing of each camera, and we do have that information. Our software is designed to so that the user doesn't have to work with that themselves. So for the user, you don't see that part of what's going on under the hood, but we do have that information. Yeah.
Okay. I think a more like software-oriented question from Olga. Is it possible to compare different ROI in a graph in real time? So I think I asked the second question. I don't know if Olga got back to us. No. Yeah, Jasper, if you have any idea.
Yeah, that's a very good question, and that's a feature request that we've been getting multiple times. So in the future update of the software, we're adding that capability. And right now, you can just select one ROI and show its information in all the graphs. But in the future, you'll be able to select multiple ROIs, and their information will be shown with the same color that's used for the outline of the ROI. So you'll be able to distinguish them very easily and compare them.
Nice. Do you have any idea when this might come out?
Off the top of my head, I think somewhere in the next couple of months.
Okay. So question from Jorge: can we get the presentation, and are you recording? So yes, we're recording. So the presentation will be available.
There's also a follow-up from Olga, I think. Is there a way around this? She's asking.
Yes.
Because before, Olga asked if you can compare multiple regions of interest. And there is a way around this at the moment. You can just export the lifetime data of an image, and then you can get it into Fiji ImageJ and do all the analysis on it there if you want to compare the regions of interest right now. So you can just export the lifetime information and do your own analysis on it as well.
A question about the camera specs from Adam Bowman. Can you comment on noise specs, dark current, read noise? Values look quite high in the spec sheets. Is it a fundamental limitation of the modulated CCD, or are there ways to improve this in the future, like deeper cooling?
That's a very good question as well. So this is a specialized sensor that can modulate at these megahertz frequencies, which is quite uncommon, and especially a sensor with those modulation frequencies and also this light sensitivity, it's not quite common. So we've been included in the development of this sensor as well, and we wanted to find the balance between light sensitivity and noise levels that go along with these high frequency modulations. So for the readout noise for our cameras, that is typically around 30 electrons, but it's below 35. So it's typically 30, and it can go up to 35. And for the dark current, we're using water cooling for these cameras to keep them at a stable temperature and to get the heat out of the camera, basically. And when we cool them, we have a dark current of approximately up to 300 electrons per pixel per second, which is quite low. We've been also looking at a lot of different cameras, different sensors that also do this, that also have a modulated sensor, and they're almost all are at least 5 times higher in the dark noise or in the dark current than this camera is.
Okay, thank you, Jasper. Another question for you from Jan. What are the upper and lower limits on lifetime that we can measure with this system? That's
a very good question as well. So for each lifetime that you want to measure, there is a corresponding optimal modulation frequency. So the trick or the rule of thumb to find the optimal modulation frequency is to divide 100 by the fluorescence lifetime that you want to measure. So if you want to measure a fluorescence lifetime of 2.5 nanoseconds, you will find the optimal modulation frequency is 100 divided by 2.5, so that would be 40 megahertz. So with our system, we can go up to 80 megahertz. So that brings you down to a lifetime of around 1 nanosecond. You can go a bit shorter, but as you go further out of the optimal range, then the accuracy will start to decrease. So that's at the low lifetime end of the spectrum. And then you can also move up to longer lifetimes by decreasing the modulation frequency. Now we can decrease the modulation frequency down to 13 kilohertz. And that gives us a lifetime range, or a lifetime in the order of tens of microseconds. Yeah, I think 10 to 20 microseconds is definitely possible with this camera as well. And everything in between as well, of course.
Good. A follow-up question on that from Neil. How many lifetime components can you discern with this system? I guess in one acquisition. And does 3 components fitting become a little hand wavy depending on how close they are?
If the three components are very close to each other, then it's definitely very, very difficult to tell them apart. Yeah, that's right.
Do you maybe have some order of magnitude of how close they can be so we can properly discern them?
Off the top of my head, it's very, very difficult to say. It also depends on the brightness of your sample and on the standard deviation on each of those lifetimes as well. So that's actually a complicated question that requires a longer answer, I think.
Okay. We can follow up on that. So question from how do you need to record reference every time you use it? Every time you, basically for every single optical configuration. So if you change the configuration in between, if you change, I guess, the objective or the filter cube or something like that, then you will want to record a new reference, and also from day to day, for instance. Am I correct, Jasper?
Yeah. Once again, you answered it perfectly. Yeah, definitely.
Question for Malte, do you need harmonic modulation, or does it also work with rectangular? I think it actually works with rectangular. Yeah.
Do you need? Oh yeah. Do you need harmonic modulation? When you're using rectangular modulation, when the output of your light source is rectangular modulation, and then I think your sensor has to match that exactly. But if you're using rectangular modulation, you get a lot of harmonics in your signal. When you're using a sine wave, you just have a single harmonic. That's your main harmonic. So you would be getting some more harmonics in your signal that would, I think, complicate your analysis.
Another question from Bo, following up, I think the answers we gave about the illumination. So even if you already have your own laser light source in your microscope, do you still need to switch to the special light laser source? So no, the laser source actually are not that special. If you send us the specs, we can tell you quickly if we can work with them. The lasers are not that special. The LEDs are more special. So if you use a regular LED source for wide field illumination, there's a high chance that you cannot use it. If you use a laser source, there is, I would say, a high chance that you can use it. So just send us the information and we can tell you for sure. We have a question from Malta. I guess it's okay. Follow up. Okay. And last question right now: what to do if the sine function doesn't fit very well? How to get better SNR? From my experience, raising the gain doesn't help with SNR. Also, what's the best range of intensity to record lifetime?
Okay, so it's a multiple part question. Let's see. If the sine wave function doesn't fit very well, so this can happen when the light intensity is very low. So when the signal to noise ratio is also not very good. And then there are several things that you can do. So one of them is increase the exposure time, which is the best way to get a better signal to noise ratio. And I agree that raising the gain doesn't help a lot to improve the signal to noise ratio. It can help a little bit. But one of the things that you can do is you can increase the exposure time. Another thing that's also very important for FLIM experiments is make sure that your biology of your sample is optimized as well. So make sure that your sample emits as much light as possible, because that will just get you much better results as well. And then there's also the question in there, what is the best range of intensity to record lifetime? The best range of intensity, well, there's no specific part of the range that you can measure, as long as you don't over or underexpose your image, I suppose. So if your light intensity is very low, if the intensity is very low, then it will be more difficult to record a lifetime image. You'll have to use a longer exposure time. And if your light is just very strong, you can use a much shorter exposure time, or use neutral density filters maybe in your microscope as well, to get the light intensity to within the range that won't overexpose the camera.
Okay, do we have, thank you so much, Jasper. Do we have any other questions? Looks looks like no so far, but again, if you have more questions in the future, please do not hesitate to reach out to us. And in any case, we'll reach out back to you with the link for the video, just in case you missed some part, and with more information. Yeah, can we talk about cost? Yes, the Toggel camera itself is it's about 80 to 90 thousand USD. And the illumination, it depends on what you get, of course. But from the top of my head, I think a four LED combiner, the multi-LED with 4 LED is about 20K USD. I think 20 or 25, something like that. So you end up with a full system for about 110, 120K. For a laser, it gets more expensive.
Yeah.
And how do you calculate FRET efficiency from the lifetime change? I think you basically like go for it, Jasper, but you compare the two lifetime before and after, and
yeah, that's right. You get the you use the equations in the FRET efficiency papers. This is also one of the features that we want to implement in the future, to build that into the software as well, to calculate FRET efficiency, because it's such an important application of the system. So that's also something that's definitely on our to-do list.
Cool. Any other question? That we're already over the hour, but we can maybe hold it 1 or 2 more minutes. Okay, it looks like we are good. So thank you so much, everyone. And we hope that you enjoyed this webinar. Sorry again for the technical issue. It's something that happened today, basically 2 hours before the demo. So really was hard to deal with. But I'm sure it was satisfying enough, and we're happy to follow up with a one-to-one demo or maybe another webinar in the future.
Definitely, yeah.
Thank you so much, Jasper, and have a good day, everyone, and stay safe.
Thanks, everyone. Thanks for joining. Bye.